Call to order the Society's 2,367th meeting in the 145th year since its founding in 1871. Good evening, everyone. Good evening. My name is Larry Milstein. I am the president of PSW, the oldest scientific society of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members and guests to tonight's lecture by Tony Zador. We'll begin with a few announcements, followed by a reading of the minutes of the 2,366th meeting and a brief recounting of the 29th meeting of the Society that took place in 1872. We will then turn to this evening's lecture, followed by a question and answer period. Thereafter, I will present a small thank you gift to our speaker, make a few closing announcements, and then adjourn the meeting to the social hour. Please join me in thanking the sponsors of the Fall 2016 and Spring 2017 lecture series, the Policy Studies Organization with the American Public University, and a generous sponsor is asked to remain anonymous. In addition, and most importantly, tonight's lecture is sponsored by PSW member Tim Thomas, who's here in the audience. So let's please thank him. I am pleased to announce that the following new members have been elected to the Society. Jennifer Weller, Associate Professor of Bioinformatics and Computational Biology at UNC Charlotte, currently on sabbatical at the National Science Foundation, who is interested particularly in genomics and computational and synthetic biology. Andrew Abraham, an attorney with the Department of Justice, with particular interests in data science, machine learning, and the intersections of science and law. And Tony Zador, tonight's speaker, whose interests will, I think, be clear from tonight's talk. Please join me in welcoming them to the Society. I should say that if Jennifer or Andrew are in the audience, they should come and see me so that they can pick up their copy of the first volume of the Bulletin of the Philosophical Society that began publishing in 1871, one of the many benefits of membership. The minutes of the previous meeting's lecture on the prospects for life on planets around other stars will now be read by External Communications Director Preston Thomas.
At the Cosmos Club in Washington, D.C., on October 7, 2016, President Larry Milstein called the 2,366th meeting of the Society to order at 8:05 p.m. He announced the order of business and welcomed new members. President Milstein shared news that PSW member and speaker Sir J. Fraser Stoddart had been awarded the 2016 Nobel Prize in Chemistry. President Milstein summarized the 28th meeting of the Society held in 1872. The minutes of the previous meeting were read and approved. President Milstein then introduced the speaker for the evening, Carrie M. Liss, Principal Staff Scientist of the Planetary Exploration Group in the Applied Physics Laboratory at Johns Hopkins University. His lecture was titled, Prospects for Life and Human Habitability Around Nearby Stars. Dr. Liss began by explaining that we used to ask whether there were planets around distant stars. We now think that every star has planets, and each likely has an Earth-sized planet. The new question is whether there could be life on these planets, what combinations of conditions might be necessary for it to develop, and how we can detect it. There are 100 billion stars in our galaxy, and Dr. Liss explained that for a star, and thus for the possibility of life around a star, mass is destiny. The greater the stellar mass, the brighter a star is, and the shorter it lives. Most stars that are visible from Earth with the naked eye are blue-white stars that burn out in as little as 1 billion years, far too short to support the development of life. The stars nearest to Earth are mostly red dwarf stars that have long lifespans but comparatively low energy, which would require planets to be dangerously close in order to derive sufficient heat. a yellow dwarf star similar to our own can be expected to last about 10 billion years. When evaluating exoplanets as candidates for life, Dr. Liss explained that an orbit within the habitable zone is necessary but not sufficient for the development of life. Factors such as the planet's orbital characteristics, tectonic activity, and atmosphere are also critical. Earth would have frozen solid but for volcanic activity, staving off a permanent ice age. Mars lost most of its atmosphere to solar wind and slowly froze. On Venus, a runaway greenhouse effect has taken its surface temperatures far outside what is suitable for life and what would be expected based on its orbit alone. Dr. Liss noted that the management of atmospheric CO2 remains critical to the continuation of life on Earth. The Earth has benefited from an extraordinary series of circumstances conducive to the existence of liquid water and the origin and continuation of life, including being the right distance from a star of suitable mass, having a Jupiter-like neighbor to scour the orbit clear of comets, a fortuitous impact by an asteroid or comet that brought water and organic materials, and a molten core that provides both a protective magnetic field and seabed hydrothermal vents that likely provided the conditions for early life. Once life has formed, Dr. Liss noted that it takes many and varying forms, most of which have gone extinct due to identifiable impact events. By sheer longevity, dinosaurs remain the most successful species on the planet, having lasted for 120 million years. By mass, bacteria are the most successful, individually and as part of symbiotic systems, which include us. In all its forms, however, we have only ever found life where liquid water was present. Thus, if we were to detect an Earth mass exoplanet orbiting a star similar to our own, the next question would be whether it has biomarkers, first and foremost, water. Dr. Liss explained that one of the most promising methods is to look for blue light spectra consistent with water, or a red edge resulting from the absorption of red wavelengths by green plants. Dr. Liss addressed the Fermi paradox, which juxtaposes the likely abundance of intelligent life in the universe with the lack of evidence for it thus far. Dr. Liss posited that the Earth may be the first planet to develop intelligent life, or that intelligent life may have investigated our solar system billions of years ago and found no signs of life. Alternatively, the vastness of space may mean that evidence of pure civilizations simply has not had time to reach us yet. He noted that the nearest stars to us are only 3 billion years old, meaning that any nearby life is likely blue-green, squishy, and not very good conversation. After the conclusion of the talk, President Milstein invited questions from the audience. One questioner asked about the alien architecture stars recently detected by astronomers, which display substantial intermittent dimming and are inconsistent with known solar objects. Dr. Liss explained that observations of the star did not show any re-emission of solar energy as thermal radiation that would be expected in the case of a Dyson sphere. Dr. Liss hypothesized that a better explanation for the available data would be debris from a massive comet or other intervening stellar matter. Another questioner asked about the TESS telescope, planned for launch in December 2017. TESS will observe nearby M and K class stars for photometric changes associated with transits by large exoplanets. Promising candidates will be more closely observed by the James Webb Space Telescope when it launches in 2018. Alpha Centauri A will be among the first stars imaged by TESS in hopes of finding an Earth-like planet in our nearest neighboring star system. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:59 p.m., President Milstein adjourned the 2,366th meeting of the society to the social hour. Temperature, 19°C. Weather, overcast. Attendance, 102. Respectfully submitted, Preston Thomas, External Communications.
Thank you, Preston. Are there any corrections or comments on the minutes? I see none, so I will entertain a motion to accept the minutes as read. Bob?
I move that the minutes be accepted as read.
I have a second? All in favor? Aye. All opposed? Minutes are accepted and will be posted in due course to the website. The 29th meeting of the Society was held on Saturday, June 15, 1872. President Joseph Henry was in the chair. The bulletin does not report the location of the meeting or the time it was held. As many of you know, Joseph Henry was the primary founder of PSW and served as its president from its beginning in 1871 until his untimely death in 1878. He was the preeminent American physicist of his time. In addition to founding PSW and serving as its first president, he also was involved in the founding of the Smithsonian Institution, serving as its first secretary, and he was involved in founding and building many other institutions of science in the capital area. Mr. Asaph Hall read a paper on the experimental determination of pi, the ratio of the circumference to the diameter of a circle, based on the principles of the calculus of probabilities. The bulletin doesn't provide any details on the paper or where it might have been published, but it's worth noting that the presentation was a follow-on to a presentation at the previous meeting by the mathematician E. Frisbee on a method to calculate pi that was accurate to 30 decimal places, which is small potatoes these days, but was pretty good back in 1872. Asa Hall also read a paper on the method of least squares, and it was published in Nature in 1872, volume 6, pages 101 and 241, and you can actually, if you can get past the paywall, go and read the paper if you're so interested. Hall was among the most prominent American astronomers of his time. He's especially noted today for measuring the mass of Mars, for discovering Mars's two moons, Deimos and Phobos, for calculating the orbits of several moons and double stars, and for determining the rotational period of Saturn. Theodore Gill presented a memoir of Mr. F.B. Meek on the discovery of new species of fossil plants from Allegheny County, Virginia, and some information on the local geology. Gill was an ichthyologist, mammologist, and malacologist. He was professor of zoology at George Washington University and served as Smithsonian librarian and assistant librarian at the Library of Congress. He was elected president of the AAAS in 1897. He was an author on over 400 scientific publications in his career, which maybe isn't so much now, but that was quite a few then. Unaccountably, at least to me, he never served as president of PSW. Fielding Bradford Meek was a geologist and paleontologist. He didn't have much formal training and acquired his skills by working more or less as an apprentice to various members of the Geological Survey on expeditions in New York, Virginia, Nebraska, Missouri, and elsewhere. Eventually, he was well recognized as an expert in his own right. He was especially esteemed for his treatises, written with Dr. Gabb, on the paleontology of California and on the invertebrate Cretaceous and Tertiary fossils of Upper Missouri country. He was somewhat infirm and diffident, and he lived much of his life in a room at the Smithsonian Castle that had been afforded to him by Joseph Henry, who was then the secretary of the Smithsonian, and who routinely made lodgings there available to working explorers and scientists like Meek, who were contributing to the Smithsonian Institution's collection and to its scientific output. Meek died of tuberculosis prematurely, as did many in those days. Finally, O. Stone presented a personal note on errors in the first survey of reference stars, which he helped produce. I have not been able to find any information on the presentation or on Mr. Stone, other than that he authored an extensive survey of double stars. And with that, we turn to tonight's lecture on understanding the brain's network using DNA sequencing, and it's my pleasure to introduce tonight's speaker, Tony Zador. Tony is the Neuroscience Chair and the Harrison Professor of Biology at the Cold Spring Harbor Laboratory. His research on cognition combines physiological, molecular, behavioral, and computational studies of the neural mechanisms underlying auditory processing, attention, and decision-making. He and his colleagues recently developed a powerful new method for using DNA sequencing to determine on a brain-wide scale the interconnections between individual neurons and to study their dynamics. Tony came to Cold Spring Harbor as an assistant professor directly from his postdoctoral work at the Salk Institute, and he has been at Cold Spring Harbor ever since then. He's an author on numerous scholarly publications, and he actively pursues opportunities to present neuroscience to a wider audience. Among other honors, Tony holds the Alla Davis Harrison Chair in Neuroscience at Cold Spring Harbor. He has been appointed a Brain Research Foundation Fellow, an Allen Distinguished Investigator, a Mathers Foundation Investigator, and a Sloan Foundation Fellow. In addition, he was named a top 100 global thinker by the global publication Foreign Policy. Interestingly, he earned a BS in linguistics at UC Berkeley and then went on to earn a PhD and MD at Yale. Please hold questions for the end, when we'll have an question and answer period, and join me in welcoming Tony to the podium.
Can you hear me? Yes. Oh, well, thank you all for coming tonight, and thanks for inviting me. It really is an honor to speak as the 2,367th speaker, and to hear about the talk of the 2,366th lecture. So yeah, I've really enjoyed the traditions so far. They're very different from the traditions that I usually hold or follow. When giving a lecture, I don't usually get to have a wonderful meal and several glasses of wine before giving a talk, and we'll see how that works out. As I understand it, the history here is that for the first few decades of this society, the rule was that they would neither eat nor drink before the talk. And I can see some logic in that, but I can see some logic in showing up a little bit loosened, shall we say, which might be appropriate for this talk. Actually, what was really appropriate was so I'm going to talk about can we upload our consciousness to the cloud, but what was really appropriate was the whole discussion about life on other planets. Because as a kid, I used to really enjoy watching Star Trek. It was one of my favorite shows. And as many of you know, that was a show about traveling to distant planets to go where no person had gone, to boldly go where no person had gone before. But one of my favorite things about that show was the transporter. Oh, I'm going to do that 6 or 7 times before. There we go. Yeah, it was the transporter. So for fans of the show, you'll know that what the transporter was a way of moving people around. And so the idea was that when the crew of the Starship Enterprise needed to go somewhere, if they were circling a planet, so they would use their big ship to go from one star system to another, but when they needed to get from their ship, which was orbiting the planet, to the surface of the Earth, they would use the transporter. And the way the transporter worked was it would dematerialize people who were standing on these little discs and rematerialize them at their destination. And as a kid, it got me, well, I mean, obviously there are all sorts of benefits for that, commuting and whatnot, but one thing that sort of troubled me was whether the person that would be rematerialized would be the same person that started off, right? So if you destroy all your molecules here and then replace all those molecules in exactly the same configuration somewhere else, are you still you? And I grappled with that quite a bit, and without any evidence for or against, because this hasn't yet been invented, I decided that yes, I would be. And so that sort of set me on my career as a physicalist. So I believe that our consciousness is somehow a consequence of the configuration of our molecules. That if we could take our molecules one by one and position them somewhere else, we would still be us, all right? So when I started graduate school, I was interested in trying to figure out what is it that is special about brains that prevent us from making computers that are as good as the ones from some of my other favorite science fiction movies of the time, either the benign C-3PO from Star Wars and the less benign Terminator. And so I spent the good portion of my PhD building neural networks, artificial neural networks, and trying to understand how artificial neural networks differed from biological networks. And my goal for a lot of that work was to actually build artificial systems. In the end, I decided to pursue at the end of graduate school, I had to make a decision. Was I going to actually try to understand a real brain, or was I going to try to build an artificial system? And I went with the biological brains, but a lot of my colleagues from graduate school actually did go on and build things like this. So many of you might have heard that Tesla is about to deploy a self-driving car that's based on the neural network technology that we were developing in the late 90s, and Google has a less stylish but apparently similarly effective self-driving car. So some of the goals of our research back then have actually been in part achieved. So much so, actually, that Elon Musk, the founder of Tesla, is actually very worried about it. So this is taken from an article just a couple months ago. Elon Musk is not alone among Silicon Valley entrepreneurs in worrying that AI will take over, and he has some plans for how to avoid the scenario. So he's not quite so worried that Terminator is going to come over, that we're going to build a vicious robot that will travel back in the future and wipe out the human race. Or he's more worried that we're going to become irrelevant, that we're going to become essentially, what does he call it? The house cat for the robots of the future. And what he would like to do, I think, is build brain-machine interfaces so we can benefit from machine intelligence. So at least some people are taking this seriously. But none of this is really the ultimate goal of a lot of these Silicon Valley magnates. What they would like to do is what's shown here in this movie Transcendence and in a lot of other movies, which is to actually upload our brains to the cloud. So what they would like to do is figure out something about what makes us us among our neural connections, among our nervous system, and abstract that sufficiently so that we could suck out the essence of us and put it in a computer, and we would somehow regain our essence in a computer simulation. And that sounds completely fanciful, and you might think that no neuroscientist would embrace that idea. But in fact, there are some neuroscientists, at least, who think that that's not unreasonable. So last year, Amy Harmon, a reporter for the New York Times, wrote an article about the neuroscience of immortality. And there are people who really are hoping that they're going to live forever by uploading their consciousness to machines. And she interviewed me and many other neuroscientists. I sort of equivocated, and I learned from this that if you equivocate in an interview with a reporter, you will not be quoted. So I spent a good 4 or 5 hours talking to her, and not a single syllable of what I said was quoted. On the other hand, a more eminent colleague of mine, the inventor of the two-photon microscope and a really well-known guy, Winfried Denk, was quoted, and he wrote, or he said, apparently, I can see within, say, 40 years that we would have a method to generate a digital replica of a person's mind. It's not my primary motivation, he goes on to say, but it is a logical outgrowth of our work. And so this is a very respectable neuroscientist, often held forth as a future Nobel laureate. And so he's gone down on the record for saying that, yes, we're just incidentally, we're going to be able to upload our brains to the cloud. So just to finish that, there's a Russian billionaire, Dmitry Itskov, who would like an even faster timetable. He would like to finish that project within the next 30 years. I don't know how old he is, but I guess that accelerated timetable is somehow related to his age. And so he is investing many billions of, or some number of billions of dollars in developing this technology. So, all right, if we're going to upload our brains to the cloud, how would we go about it? Where is the essence of us? Where is the we that we would do the uploading from? And this is really just to frame that question of finding what is it that is the kernel of our consciousness. Where does it reside? I don't know the answer to what it is, but I think I know where it is, and that's what I'll be trying to talk about. So I think it's, and I think the field thinks that it's all in the circuitry. So I thought it would be useful to go back to Descartes, who some 350 years ago, even before the founding of this philosophical society, published what is probably the first suggestion for a circuit that describes a human behavior. And so he was interested in the circuitry by which we withdraw our foot from a fire when it's there, because evidently that's an important thing to do. And so I guess he camped out a lot or was clumsy. So the mechanism that he proposed was that the flames of the fire displace the skin, which pulls on a tiny thread. The thread opens a pore in the ventricle. The animal spirits flow from the ventricle F through a hollow tube, and then the animal spirits inflate the muscle of the leg, causing withdrawal. So I know that some of you out in the audience are MDs, and you may recognize that this is not exactly how we think it works today. We know, for example, that the animal spirits are stored a little bit more forward here. No, but seriously, I do really like this mechanism because it is a mechanism. I think it is really the first time. So of course he got the details wrong, and there's a good reason for that, was that he didn't actually do experiments. He was doing science at a time before there was the idea that you should come up with a hypothesis and then go about and test it. But he did have a mechanistic explanation that at least had the right form from my point of view. In principle, you could imagine building a machine that operated according to these principles if you could indeed isolate the animal spirits. So that is, he did get the idea of a reflex right. Today, we still believe in reflexes. Most of you have been to the doctor and have had your knee tapped. And the way that works, that's a reflex. So you tap your knee and your foot goes forward. And why does that work? That works by a reflex. Essentially, there's when the hammer taps the patellar tendon, that activates a nerve that sends a signal back here to the spinal cord that forms a connection, a synapse with another nerve. I'll get it. It's probably not a problem they had back in the 1800s. That forms a connection with this one and activates the muscle here and stimulates this muscle to contract. And all that happens in a fraction of a second, and that's why your leg moves. So that is a circuit, and we can understand that circuit. And ultimately, the idea that neurons are connected to each other and cause stuff to happen, right, is the basis for systems-level neuroscience. So what I spend much of my time working on at the lab is trying to understand how we make decisions. All right. So when I got to Cold Spring Harbor, I was interested in how we make decisions about sensory stimuli. So we imagine that you receive some kind of a sensory stimulus, a sound, and you have to make a decision about it. So depending on what exactly you hear, you have to perform an action. And I was interested in understanding what is the circuitry that allows you to convert a sound into an action. So if I say right, you might raise your right hand. If I say left, you raise your left hand. So in that sense, you've converted a sound into an action. So that's hard to study in humans. And traditionally, the preparation, the animal preparation that people studied this kind of decision-making in was non-human primates, macaque monkeys. And when I got to the lab, along with a former colleague of mine, Zach Mainen, we decided that it would be really useful if we could study that kind of sensory decision-making in a rat. And the reason we wanted to study a rat was that it was already clear 15 years ago that the tools that would be available for dissecting the circuitry of rats and mice would advance much more quickly than the tools for dissecting the circuitry of the traditional decision-making preparations, namely the monkey. So what we did was we figured out how to train rats and later mice to perform tasks like this, and I'll just give you a flavor of this task. So what you see here is a rat. It's sticking its nose into one of 3 ports. There's a computer so there's a little light beam in here. When the rat sticks its nose into the center port, it breaks a little light beam that sends a signal to the computer, and the computer then delivers a stimulus. And we've trained this rat so that when it hears a high-frequency sound, it should go to the left, and when it hears a low-frequency sound, it should go to the right. And it's a thirsty rat, and each time it gets the right answer, it gets a little water reward. So it will do this 1,000 times in a row. And I'll just show you a little video of this. It's a little loud, sorry. So it could go on like that it will go on like that 1,000 times or more. So what I hope you see is that we've converted the rat here into a little machine, almost like a reflex, but it's like a programmable reflex. It's a reflex that is quickly and dynamically configurable from one thing to another. In this particular case, it's a reflex that converts sound into an action. And so and we have many other tasks. This is an auditory version. There are lots of twists and turns. My former colleague Zach studies olfactory decisions, and other people study visual decisions, and we've discovered all sorts of things. But ultimately, what we're interested in doing is bringing that down to the level of circuitry. And so after we'd been working on this for about 10 years, we sort of began to realize that although the tools were largely available for us to test any hypothesis for what the circuitry might be, we didn't have a clue what the circuitry was. We didn't have a clue as to what the wiring diagram was, which would then provide a foundation for allowing us to relate the circuitry to the actual behavior. And so why might you want to know the circuitry? Well, so one reason is to understand basic decision making, but another reason is that many neuropsychiatric disorders actually arise from disruptions of circuitry. And I think so here are some disorders that might arise in part or in whole from disruptions of circuitry. They include autism, schizophrenia, depression, addiction, bipolar. And I would say that our lack of understanding of this circuitry has stalled any kind of progress, or has largely stalled most progress in developing treatments for these disorders. So for the last 10 or so years, pharmaceutical companies have largely given up trying to come up with new treatments. So there was a wave of drugs that came out, and then a wave of profitable Me Too variants of those drugs, but no drugs, or very few drugs, that address fundamental new mechanisms have really come out in the last decades, to the point where major pharmaceutical companies have largely shut down their research divisions and basically punted and are waiting for us basic scientists to make advances. And I think the reason is that the traditional kinds of approaches to understanding neuropsychiatric or to addressing neuropsychiatric disorders lack circuit specificity. And this is a cartoon and an exaggeration, but the cartoon is that basically they consider the brain as brain soup, and they imagine that if you bathe the brain in some particular drug, it will change the overall organization or activity of the brain. If you bathe it in another drug, it'll change it in a different way. So there are drugs that treat schizophrenia and drugs that treat depression, but they largely lack circuit specificity, and to the extent that they work, it's because they've tapped into some degree of circuit specificity. Really, I would say that many of the major advances over the last decade have come not from pharmaceutical approaches, but from approaches that actually target specific neural circuits with deep brain stimulation. And so deep brain stimulation is increasingly used to treat intractable conditions like Parkinson's, chronic pain, epilepsy, depression, Tourette's, obsessive compulsive disorder. So I'm not going to be talking about diseases tonight, but that's sort of the practical aspect of what we're doing. But ultimately, it boils down to the fact that what we need is to understand the brain's wiring diagram, and we need to understand it at sufficiently high resolution that we know what to manipulate, what to modify, and how the various aspects of the circuitry relate to behavior. So you might ask, if you follow the New York Times, don't we already have the wiring diagram? So with some fanfare over the summer, a multi-year, multi-center, multi-million-dollar project headed by David Van Essen was released, and the title in the New York Times was "Updated Brain Map Identifies Nearly 100 New Regions." All right. So, and I don't mean to say that this was not useful, but I would say that this is a very low resolution. This is the Human Connectome project. The data revealed 97 new regions, bringing the total to 180. So this is a very low resolution map of the human brain. So it's not the wiring diagram. It's a very crude description of the overall organization of brain regions. We have a similar kind of description, low resolution description of the mouse. Most recently there was the Mouse Connectome Atlas was published a few years ago and is available online, an incredibly useful resource for my lab and many others by the Allen Institute, founded by Paul Allen, the co-founder of Microsoft. It took a few years and a few tens of millions of dollars to put this online. And I'll just say here as a digression that most of the work that I'm going to be talking about today is in rodents, rats and mice. And you might wonder, okay, why are you working on rats and mice? What relevance does that have to humans? And I would say that it is there's a lot of evidence to support the idea that the basic organization of the cortex has been preserved across mammals. And so what you can see here is the cortices of a half dozen different animals: mouse, rat, rabbit, goat, cat, cheetah, lion, dog, and human. And I assume they all look the same to you because they all look the same to me. There are experts who know more than I do about this kind of anatomy to whom they do look different. But for me, the main thing that I see the main differences I see between these different cortical areas is that they're of different sizes. But essentially, for example, it turns out that there are the same number of neurons in each one of these columns. And they're organized, as far as we know, in largely the same way. So there will surely be differences between mouse and humans, but so far we haven't seen much evidence for many differences. And we think that we'll be well on our way to understanding the human cortex and the human brain if we could understand the mouse brain. So they're similar except size, and just to remind you, this is one reason that we find it easier to study the mouse than the human because it's a lot smaller. All right, so what do we have? What do we actually know about the wiring diagram of the brain? I would say that roughly speaking, what we have is a map that looks like this. This is a map of the major interstates in the United States, and what we would like is something more like this. If you actually want to find your way around, you need to be able to have a high enough resolution picture to find out how to get to the Cosmos Club and where the parking lot is. All right? So why do we not have, what is the limitation, what is the challenge in getting a high resolution map of the nervous system? Well, I think this picture pretty nicely sums it up. This is a high resolution view of the wires emanating from a few dozen, from maybe a dozen neurons in the cortex. So each one of these little things is a neuron, and these are the dendrites that come up. They receive the inputs from other neurons, and the rest of these wires are probably mostly the axons which send out the signals from other neurons. And so each one of these neurons is receiving input from thousands of other neurons, and it's sending its axons both locally and long distances. And what you see here is that it's a tangled mess. What you see is that if you would like to know where this neuron's axons go, you might try to trace it by looking really carefully, but as soon as you start looking carefully, you can see that you can't see where it's going. And that is essentially the fundamental problem of trying to trace neural circuits at high resolution. It's the fundamental problem of trying to figure out where neurons send their information, is that it's physically just very difficult to trace those little axons. And so what are the alternatives? Well, one alternative is you might decide that you're not that interested in where each individual neuron sends its information. You might decide that it's good enough for you to know where, on average, all the neurons here send their information. And so that's one common approach, and that's the approach that was used by the Allen Brain Institute and many other mapping studies, and most of the mapping studies that are done, is that if you're interested in where neurons from a particular area send their information, if you were interested in where the neurons, so each one of these little green balls is a cartoon of an individual neuron, if you want to know where the neurons here send their information, you inject some kind of a tracer that labels the cells green, and you wait for the green to spread to the distant axons, and then you can dissect out the individual target regions. All right? And so if you inject the tracer in a particular place and you see green somewhere else, if you do the experiment right, you know that the green got to one of those target regions because you put it here. And so you know that if there are 5 candidate target regions and you see green in 3 of them, you know that neurons in that area sent their information there. And that's super useful. But what you don't know is whether the individual neurons are organized like this, so each neuron sends its information to one place, or maybe each neuron sends its information to all 3 places. Or after years of working in biology, the more likely, almost inevitable conclusion is that it's complicated. That some neurons send their information to one place, some neurons send their information, and there are unexpected configurations always. So if you actually want to know how information is routed through the brain, you can't just use these bulk tracer injections. All right? You can't just label a population and count on that to give you the information you need. So what's the alternative? Well, there's so to drive that point home, actually this was a story that was told by my graduate student, Eustace Capsule, who is really the driving force. He's a force of nature, and he's the driving force behind this work. And he works very hard, but he does occasionally take vacations, and he's a spontaneous guy. So his thought was, okay, I'm going to go on vacation. I'm just going to drive to the airport, and I'm going to hop on a plane and go wherever the plane, the first flight takes me. And so we live in the New York area, and he's from Germany and really doesn't know the area that well. So he drove to LaGuardia. So for those of you not familiar with LaGuardia, there are a couple things to note. First of all, it looks like a third world bus station, which I think is an insult, apparently, to third world bus stations, I've been told, for people who travel in the third world. But the other really disappointing thing for Eustace when he got to the airport was he discovered that LaGuardia is only a domestic airport. And so he had the option of going to Toledo or Cincinnati for his, I actually don't know where this one goes, so he might have taken that one. So the point is that in this analogy, the airport is like a neuron, and it's really important to know where the neuron sends its information. It's really important to know where the flight routes of a given airport are. So if you really want to know that in the brain, what would you do? Well, there is a time-honored classic neuroscience technique for figuring that out. It's called single neuron tracing. It's an art form. It's very difficult. It requires taking a fine glass pipette and bringing it up onto a neuron and then targeting just one neuron and filling it with some kind of a tracer. And then waiting for that tracer to spread throughout the brain and then dissecting the entire brain looking for places where that neuron sends its projections. And so this is a wonderful paper that actually my lab relies on quite a bit. But from the point of view of this discussion, the relevant thing is that this paper describes the projections of 25 neurons from 28 rats, because you can only do one neuron per rat, okay? And as a reminder, as my advisor used to begin, my postdoc advisor used to begin his public talks by pointing out that the human brain has 100 billion neurons connected by 100 trillion synapses and over 100,000 miles of axons and dendrites. And we in neuroscience call that job security. And this is why it's job security, because with the 100 billion neurons, there aren't even enough rats in New York if this is going to be your approach to figuring out the connectivity of individual neurons. So, all right, that's not quite the state of the art. There was a very clever idea that is actually the inspiration for my work that came along about 10 years ago. And I believe Jeff Lichtman spoke here a few years ago. He probably talked about his electron microscopy work. But another really fantastic thing he did was he invented, along with Josh Sains and Jean Livet, a technique called Brainbow. And it's incredibly clever, and it was the inspiration for my work. And in that work, what they did was they made genetically engineered transgenic mice, which they tricked. They embedded a little piece of DNA into those mice, into every cell of the mouse. But that little cassette of DNA would be subject to a random scrambling after the neurons were born. And it would be scrambled randomly, differently in each individual neuron. And the way that cassette was designed, it could be scrambled in as many as 200 different ways. So the cassette consisted of a bunch of different colors. And you can imagine those as a bunch of playing cards. And in some cases, the cards were face down, in which case you wouldn't see the color. And in other cases, the cards were face up. And because of the way that cassette was designed, they could be, it's like you could shuffle the cards and turn them up and down in as many as 200 different configurations. And it produced really beautiful pictures like this one. And you can see that if the axons have different colors, then it's a little more straightforward to distinguish them as they course throughout the brain. So theoretical diversity of 200 colors. The problem was that in practice, you can't distinguish anything like 200 different colors using any kind of modern microscopy. In practice, you can't really distinguish more than about 3 or 4. So on the one hand, it was inspirational and incredibly clever. On the other hand, from a practical point of view, it wasn't really that useful. And that's really sort of driven home by this. It's almost certain that these 3 purple neurons are different color purples. But not only can you not distinguish their purples on this slide, you can't distinguish them with most microscopes. So when I saw his talk about 6 or 7 years ago now, I got to thinking, well, why use colors if colors are so hard to distinguish? Why not come up with something that's not so hard to distinguish? And I went through like a couple weeks thinking about quantum dots and all sorts of, really what it has to do with is the fact that the color spectra of these genetically encoded indicators are quite broad, and had the idea that, well, quantum dots have very sharp ones, and if you could encode specific sequences within the neurons that would bind to specific quantum dots, then maybe you could distinguish more quantum dots. And then when I got thinking about that, I realized, forget about the quantum dots. Why don't we just read out the sequences? We have a great technology for reading out sequences, and it's called sequencing. And so that was basically the eureka moment. It's probably the only eureka moment I've had in my life, or at least in my career, where I said, why not use sequences instead of colors to label neurons? And so this slide, so as most of you know, our genetic material consists of strings of 4 nucleotides, ATGC. And if you put them together in different combinations, there are a lot of possible combinations of ATGC. And so the idea was that if we could endow neurons, if we could make neurons express random sequences of RNA, then each one would have a unique label. And so that is the origin of the technique that we now call MAPseq. I'm not giving you the acronyms of the 2 or 3 or 4 failed earlier versions of it, and this is the version that actually works. And so basically the idea is that MAPseq is, if you like, a digital rainbow, where we replace colors with numbers, where the numbers correspond to the individual sequences. So each neuron will express a unique sequence of DNA, a genetic barcode. And the theoretical diversity is effectively infinite. So that's, I'm going to say it again, that's the core idea. If you're going to just go back to sleep in a minute, just wake up briefly, because that's really, if you can remember this, you can sort of piece together the rest of my talk, which is the core idea is to recast connectomics, neuronal connectivity, as a problem of high throughput DNA sequencing by labeling each neuron with a unique sequence, a DNA barcode. All right, so if you guys want to go back to napping or checking your email, now's the time. But that is the take-home message. So why sequencing? Well, 2 reasons. It's cheap and it's fast. And for many of you, this will be familiar, but for those of you for whom it isn't, it's interesting to see. So this is a plot of cost versus time starting in 2000. And this white line here is Moore's law, the rate. It's a logarithmic plot, so this is 10,000, and this is fractions of a dollar here. And this is, Moore's law is the rate at which computers have gotten faster and cheaper over the last 40 or some odd years. It's an exponential growth. It's incredibly fast, right? It's the reason that my iPhone has more computing power than the top-of-the-line computer that I did my PhD thesis on, right? So it's a very respectable rate of growth. But what's truly impressive is that drawn here is the cost of sequencing. So the human genome cost about $1 billion to sequence in 2000, and the cost dropped following Moore's law up until about 2007. And then, as you can see, it just took off. I mean, the cost just dropped at a rate that was absolutely incredible, right? It was much faster than Moore's law. And if you extrapolate this out for the non-mathematically inclined, you can see that approximately around the year 2019, it will cross 0, and they will actually pay for you to. So obviously, no, it's a logarithmic rate. That's not quite true, but there is truth to that, right? Because the value of your genome will be such that the cost of obtaining your genome will be trivial compared to the value that your gene sequence actually has. So the idea that we have is that if we could piggyback off of this incredibly impressive technology, if we can convert the problem into connectomics into a problem of sequencing, then we're golden, right? This is one of the few sort of things that scales even better than, that is on the right scale of numbers of neurons, which are 10 to the 7, 10 to the 8, number of synapses, which are 10 to the 10, right? The number of nucleotides is not that much more, or the number of synapses is not that much more than the number of nucleotides. And so, okay, so what's the idea? The idea is that we should be able, that we're going to label each neuron with a random sequence. How many random sequences are there? Well, there are a lot. There are 4 possible random sequences for a barcode of length 1, right, ATGC. There are 16 possible barcodes of length 2, right? It could be A in the first position, any of 4 things in the second, T in the first position, any of, and so on. So in general, it's 4 to the n, where n is the length of the barcode. And even very short barcodes, so a string of 30 nucleotides is short for a molecular biologist, but even a string of 30 nucleotides has a potential diversity of 10 to the 18, which is a really big number. In fact, I looked it up the other night, and it turns out that that is approximately equal to the number of grains of sand of all the beaches on the Earth. Not sure if that helps anyone. It might be easier to just say, oh, 10 to the 18, but apparently some people like to, and so sometimes I show a picture of a beach, just to relax you. So the question is, can we label, if we start labeling neurons randomly, what are the chances that 2 randomly labeled neurons drawn from a diversity of this size will have the same barcode? That turns out to be a well-known problem called the birthday party problem, which some of you may know has this sort of counterintuitive observation. So the chance that somebody has exactly my birthday is 1 in 365, but the chance that any pair of people have the same birthday is actually much, much higher. And in fact, I believe there are more than 23 people in the audience, so it is quite likely, there are probably at least 50, so there's a very high probability that some pair of you will have the same birthday. And in general, as you'll see, it's really bad for us if 2 neurons share a barcode. So we were up against this. We wondered whether this was going to screw us over. This was going to make our life more difficult. And the answer was no. It's equivalent, once again, one of these like crazy analogies. It's equivalent to the probability. It's the fraction of the fraction of New Yorkers, because there are about 10 million New Yorkers and about 10 million neurons in the mouse cortex, the fraction of New Yorkers that were born exactly the same picosecond, because there are about 10 to the 18 picoseconds in a year. So my intuition doesn't work out that way, and so I worked it out on a computer, and it's 0. It's like 10 to the minus 15 or something. So basically, the number 10 million is a lot of neurons, but 10 to the 18 is way more possibilities. So the chances that 2 things share a barcode are infinitesimal. So we're kind of okay there. As you'll see, this is the theoretical diversity. In practice, our diversity isn't quite that high, and if I'm just putting this as a technical asterisk. If anyone actually wants to talk about our actual diversity and where it arises, I'd be happy to cover that in the question period. So it's really not 10 to the 18. It's considerably smaller, but we're careful to make sure that this probability is very low. So how does MAPseq work? All right, here's how it works. We have a bunch of neurons in a source area, and we want to know what their projections are to a bunch of different targets. So we make a viral library. We make a tube that contains a bunch of viruses that are all identical except for a 30 nucleotide random barcode. So as a side note here, viruses are little packets of DNA or RNA encoded in a bit of protein that have basically evolved and are efficient ways of sort of delivering DNA or RNA to a cell. And there are a couple of viruses that have been, so viruses you think of as being unhealthy or bad for you, but with genetic engineering, a handful of viruses have been modified so that they're not harmful to cells, so that the key pathogenic components have been stripped out, and then the space that used to be occupied by the pathogenic components or other components is now available so that we, molecular biologists and neuroscientists, can deliver cargoes of interest. And so that's been a sort of useful technology. It's the basis of gene therapy for a while. So we used a particular virus called Synbis, which operates very quickly and has a pretty high payload and expresses at high levels. And we engineered it so that it expresses, each little virion here expresses two things. It expresses a specially engineered protein that we developed that grabs the barcode, the RNA barcode, and drags it throughout the neuron and out to the distant parts of the axon. Because in general, if you have a piece of RNA in a neuron, the RNA sort of usually stays near the cell body and doesn't spread. So what I just summarized was 2 PhDs. And about 6 or 7 person years of hard work. But it works. So we have now a tube of this virus, and we inject a little bit of that virus into the brain. It infects a bunch of neurons. The neurons take it up, and then they get full of the particular barcode. So in this case, just for convenience, blue is barcode number 12, green is barcode 42, and red is barcode 712 out of the 10 to the 18 possible barcode sequences. And so the idea is that the barcode that was here, 712, is also out here, but it's not there or there, right? Because this neuron doesn't project to those two targets. And the barcode 42 is at all three places because this neuron happens to project all three, and barcode 12 is up there. So the barcodes fill the cells. Then we dissect out the brain regions of interest. So we dissect out the source and all the targets. And then we organize our data and collect it. And so when we're done, we have a simple table or a matrix that gives us the connectivity of all the neurons in the source area to all the different targets. So target A contains these two barcodes, target B this one, and target C these two. And of course, we could organize it the other way so that we could figure out that barcode, i.e. neuron 42, projects to A and B, A, B, and C, and so forth. Okay? So this matrix basically is the condensation of one of, it summarizes what we get out of one of those experiments. All right, so there are some technical concerns that some of you might be wondering about. If you're not wondering about them, then don't worry about it. But you might wonder, for example, what happens if two viruses expressing two different barcodes infect one neuron? Well, so in that case, we get a double infection. So we get, this yellow isn't showing up, but there's supposed to be a ghost neuron back there. And from our point of view, what we see is a sort of ghost neuron that has exactly the same connectivity as or a pair of neurons that have identical connectivity, arising from the fact that basically there are two barcodes, not one, in this neuron. And so I've called that 42A, all right? That's not so bad. We're interested in getting the connectivity of hundreds or thousands or millions of neurons, and we believe that they fall into patterns, and if we see one of them twice from time to time, as long as there aren't biases as to which neurons get doubly infected, it's not really a big concern. On the other hand, this would be really bad if we use the same barcode in two neurons. So if instead of labeling this neuron, this red neuron as 712, we labeled it as 12 also, then we would see one blue neuron that projected to two places instead of recognizing that these two connections come from two distinct neurons. And that's why we're very careful to avoid these double infections. All right, so what can we use this for? Well, our first application of this technique MAPseq was to a brain structure called the locus ceruleus. The locus ceruleus is a neuromodulatory, it's a neuromodulatory brain center. There are maybe I half dozen or a dozen of these neuromodulatory centers that broadcast signals throughout the brain and are thought to sort of affect brain state. And the locus coeruleus in particular releases a neurotransmitter called noradrenaline. It's the sole source of noradrenaline to the cortex. Noradrenaline is a sort of alert signal, a wake-up signal for brain circuits. And the traditional view of how the locus coeruleus, and in fact a lot of different neuromodulatory areas, but in particular locus coeruleus works, is that it broadcasts a signal throughout the brain. So basically the idea is that the traditional view has been that when the locus coeruleus is active, it sends a wake-up call to the entire brain, okay? Like you probably had you might have had a little burst. That would be mean. But instead, there's another possibility which had not been tested, which was that maybe individual neurons in the locus coeruleus could be active at specific times and could target particular brain regions. So maybe instead of delivering an overall wake-up signal to the entire brain, maybe the locus coeruleus could selectively activate, let's say, the visual cortex or the olfactory bulb, and thereby provide a more specific and selective signal. So you might imagine that if you needed to ramp up your visual acuity because you're about to hit a baseball or something, maybe your visual cortex is getting amped up with a little locus coeruleus, and you don't even want to be hearing what people are telling you. All right? So you wouldn't want to amp up your auditory cortex. But if every neuron projected to if every locus coeruleus neuron projected to every brain region non-specifically, then that just wouldn't be possible, right? So we wanted to know whether it was even possible to have this organization. We knew that if you inject in a bulk way the locus coeruleus, the projections go throughout the entire brain. And so hypothesis one is that the neurons in the locus coeruleus all sort of project widely throughout the cortex. And model two is that the neurons in locus coeruleus have very targeted projections. All right? So the experimental design is very straightforward. We injected the MAPseq virus into the locus coeruleus of a mouse. We waited 2 days for the cargo to be expressed, for the barcodes to be expressed by individual neurons, and for them to be carried out to the axon terminals. And then we sliced the brain, and when I say we, I mean Eustace, sliced the brain into 22 coronal sections, and from each coronal section, we removed the cortex. And then we collected all the barcodes that were in each of those 22 cortical sections on one side separately from the other side. So what did we find? Well, this is the projection pattern of one neuron that we found, one barcode, barcode 28. So as you can see, it's not going, so here is position along the back front axis, right? So if I align this, this would be here, this would be there, and so forth. And here is the strength of the projection in units of barcode molecules, which are basically how strongly, how much axon there is there. And what you can see is that this particular neuron projects very strongly to a point right around here and pretty much doesn't send its axons anywhere else. All right? So this is consistent with the model I showed earlier where there's targeted projections. And we saw a lot of neurons like this. But we also saw neurons like this, which were more consistent with the traditional view, which is that the neuron projected pretty much everywhere. And we saw things that were even more complicated, like this, so two-humped camel. And we saw all sorts of things. And so in the end, what we had was 1,000 neurons, and each one had a unique projection pattern. And our job security remains intact. I'm not really sure. All I can say is that it's complicated, but it's out of all the possible complicated configurations, it's one particular complicated one. And now actually our collaborators on this are using this as the foundation for further experiments to tease apart the role and function of the locus coeruleus. So it ruled out all sorts of possibilities for how it might operate, and it included all sorts of possibilities. So we're not done, right? We didn't finally say, okay, now we understand how the locus coeruleus works, but it made our lives as experimentalists a lot easier. So both models were wrong. And in fact, it's idiosyncratic with some neurons doing this and some neurons doing that. So the primary interest of we worked on the locus ceruleus because it was a really beautiful test case. There are only 1,000 neurons. They project throughout the brain. I had a colleague next door who was interested in it. I thought it was an interesting problem. But that's not my own lab's main interest. My own lab is mostly interested in the cortex, the seat of cognition. And what we'd really like to understand is how those neurons are wired up. So what I've told you so far is published. Now I'm going to tell you some work that's unpublished, but we're very excited by. So what we'd like to do now is map the connections of the entire cortex of a mouse in a single experiment, all right? And so instead of making a single injection into a mouse, one of my postdocs figured out how it's possible to make 600 injections in a single mouse. And then we dissect out the brain at much higher resolution using laser capture microdissection for the aficionados in the audience into currently about 300 different pieces. And from each one of those 300 different pieces, we extract all the barcodes. And so from that, we have a map of the connectivity of the entire cortex that has a resolution given by each one of these. This is a cartoon, but it's about right. There are about a half dozen pieces for each coronal section. And in the future, we can go even higher. So we have a map that's fairly high spatial resolution and single neuron connectivity. And so what does that look like? So this is each one of those green balls represents one of the areas from which we extracted the barcodes. And the lines represent a handful of the strongest connections on average. Each one of those balls represents about 1,000 neurons. And so it's not a good way to analyze the data, but it's a good way to sort of get an appreciation for what it is we have. We have the connectivity of actually 50,000 neurons in a single mouse. And so this is the 50,000 neurons mapped onto the cortical, onto the brain of a mouse. And for each one of those neurons, what we can tell is where each of those neurons connect. So we picked 2 of these at random, 2 out of those 50,000, and we picked a handful of the strongest connections of those 2 neurons throughout the brain. And so we now have, we've done this was an early experiment. We're doing this experiment over again, but we're now struggling with how do you make sense of data of this size? I mean, it's actually, in terms of the amount of data, it can be emailed back and forth, but the richness of this data set is unprecedented. And we're trying to figure out sort of how to think about it and what to do with it. So I'm getting close to the end. I just want to maybe share one more tantalizing future direction. So MAPseq, although it's 50,000 neurons, the spatial resolution is still not quite what one might want. One might want to know where every single neuron and every single synapse is. We might also want to know, to be able to relate the connections to recordings of those neurons in behaving animals. And so to address these and a bunch of other problems or limitations of the current technology, we're moving to a next generation version of this based on a technology called fluorescent in situ sequencing, or FSSEQ. And so I'll just take a minute or two to tell you about that. So to understand FSSEQ and in situ sequencing, I have to sort of comment about how conventional sequencing works. So so-called conventional next-generation high-throughput sequencing, such as Illumina, it works like this. You take a little tube of DNA, you chop it into little pieces. Each of those pieces, you flow it onto something called a flow cell, and it gets captured by these teeny little microscopic complementary posts. And then each one of those things gets amplified in place. And so instead of having 1 copy, you now have 1,000 copies of it. And then you add colored fluorophores, where each of the colors corresponds to a nucleotide. And you do it in such a way that each spot gets only 1 added nucleotide at a time. And so if the nucleotides have 4 colors corresponding to the 4 bases, yellow, red, blue, and green, ATGC, and so you get a picture, right? So you get a picture of a bunch of dots. And if you're sequencing 100 million pieces of DNA at a time, you get a picture that has 100 million dots on it. And then you wash away those dots, you add 1 more nucleotide, and you get a picture of the dots in the same place, but now they have different colors corresponding to the next nucleotide in the sequence. And if you want to sequence 100 nucleotides, you have to take 100 pictures. And so that's shown here. So this particular piece of DNA has a sequence C, T, T, G, or something like that, and so on. So most people who use high-throughput sequencing don't actually think about the guts. They don't think about how it works. They give a sample of DNA, and they get back a sequence. And in fact, I was like that too for the first few years, until I met a guy named Jay Lee, who is a postdoc with a guy named George Church, who's one of the fathers of high-throughput sequencing. And they realized that you don't have to extract the DNA from the brain. You can leave it in place. So this is just summarizing. Illumina sequencing works by imaging. So you don't have to extract the DNA. You can leave it in place. And if you're clever about it, and here I'm summarizing Jay Lee's 10-year postdoc, you can leave the RNA in place in a neuron, and then, so they did it in non-neuronal cells, and we've adapted it to neurons. You convert it into DNA, then you amplify that DNA in place, and now you've converted it to a previously solved problem. You now have a bunch of dots that need to be imaged, and you can use exactly the same chemistry to do the sequencing. And so that's what PSW is. PSW is still an imaging method that differs only from conventional sequencing by the fact that the thing that you're sequencing is not been removed from the cell. All right? And so we have adapted that for our barcodes. It works extremely well. And here's an example of 10 nucleotides in a plate of neurons. And what you can see, so each of these neurons is filled with a barcode, and we can sequence those barcodes in place. So this particular barcode, which is right, or neuron, which is right there, has the sequence G, you see it's green, T, it's yellow, G, it's green again, and so forth. And so for each of the neurons on this plate, we can actually get the sequence. And we're now doing that in my favorite area, which is the auditory cortex, where we can sequence a few thousand neurons and figure where each one of those neurons project. So, all right, why is this useful? Why is all this useful? Well, one application is and others are studying mouse models of neuropsychiatric disorders like autism. So we know some of the genes that cause disorders like autism and schizophrenia. We can express those genes. We can make mice that have those mutated genes. And in some cases, we have a mouse that has odd behavior that might be kind of like the behavior that you would expect a mouse that has a human disease like autism or schizophrenia to have, but we still don't know what is wrong with that mouse's brain. We think it has something to do with wiring, but we don't know what is going on. And so what we're hoping is that with this high-throughput MAPseq approach, we'll actually be able to get the complete wiring diagram of a normal mouse and then ask, how does the brain of a normal mouse differ from the brain of a mouse with a gene that might give it miswiring consistent with autism or schizophrenia or any of these other disorders. We're also using it to, we're also combining it with functional imaging of neurons when they're still in the brain. We record the activity of neurons, and then we find those same neurons and after the end of the experiment and figure out where those neurons sent their information. Okay, but this whole talk was supposed to be about whether we'd be able to upload our consciousness, and I haven't really shown how to do that. And the bad news for those of you who were hoping that we were going to be able to have a little booth at the edge there is that I actually see a pretty serious problem. I think that we're going to be able to understand neural wiring well enough to understand how mice in general work and how humans in general work. But the specifics of what makes you, and me, the collection of our memories, the way I think about it is this is a Fiat like my first car. This is a Fiat 128. My first car was not this particular one, but I found a picture of something that looked kind of like my first one off the web. And my first car was kind of a clunker. It made all sorts of funny noises. It had a gas tank that it could only hold 3 gallons at a time, which led to all sorts of challenging experiences. And it had squeaks and aches and pains. And basically, it had character. It had personality. And if I sort of liken myself to a 50-year-old Fiat 128, and if I felt some attachment to that particular Fiat 128, I might be tempted to take it apart and then replicate each one of these pieces, right, to like super high precision. But I suspect that no matter how much money I gave you, you would not be able to take apart my old Fiat 128 and copy all those pieces in just the wrong way that when you put them together, it had exactly the same flaws, personality, that my Fiat 128 originally had. And so my own hope for achieving immortality is sort of the more traditional one through publishing papers and raising children. And so I'll just close with a fortune cookie that I found. I really got this fortune cookie the night before a talk I gave a couple months ago. If the brain were so simple we could understand it, we would be so simple we couldn't. I'm not really sure what that means. And so I ponder it a lot. So with that, I'd like to thank my Collaboratome. In particular, the driving force behind this was Ian Pycon, who's a brilliant graduate student who pioneered all this work. When I talked about the 3 or 4 years spent developing the technology for that, that was Ian's work. He went on to work for Google and then now is working for a startup in New York called Calliope. And a current graduate student in my lab, Eustace Capsule, who's just a force of nature and did that work and has pretty much everything else I talked about. And also I want to thank my Fundosome, the Simons Foundation, IARPA, Sports Foundation, Paul and Family Foundation, Brain Research, NIH, and Cold Spring Harbor. And thank you for your attention.
So we have time for questions, and there is a procedure for questions. There are some microphone runners in the room, and if you'll raise your hand and keep it up long enough for them to see you, eventually a microphone will come to you. And when it does, and it's your turn, please stand up, tell us your name, tell us if you're a member, and then ask a question.
So these are presidential debate rules?
Yes, and I'm a very fair moderator.
Yeah.
So we'll have our first question over here from our evening's lectures sponsor. Could we have a glass of water up here?
My name is Timothy Thomas. I'm a member of the Society. You started your lecture by saying you had worked in neural networks, which are typically used to do brain-like and crude way computation. Is there anything in your findings which can be used to do brain-like computation? Or does it be so messed up by temporal issues, chemical issues, inhibition issues, and all sorts of issues?
That's a great question. So I started my career in neuroscience with pursuing the idea that the difference. So I started at a time when I could confidently say that there are all sorts of things that brains can do that humans can't, like drive cars and do translation and all sorts of other things like that. And that list of things is getting shorter. I still believe that there are huge fundamental stylistic differences between how brains work and how machines work. When I started, I thought that the key differences were in the individual processing units, that the way that artificial neural networks work is that they're basically summation nodes followed by a threshold. And real neurons are far more complicated, and I spent a lot of time studying the nuances of individual neurons, and there are differences, but in the end, I concluded that that was not the key difference, that you could replace one biological neuron with 10 artificial neurons and it would be fine. And that I now have come to the really unfortunate conclusion that what makes us better, I subscribe to what's called the bag of tricks model. What makes us better is that we've benefit from several 100 million years of evolution, and we're born with neural networks that are wired in just the right way to solve the problems that we need to solve, sort of by definition. And the problem with with artificial neural networks is that typically what they try to do is they try to boot up from scratch. And I've had this debate with people who work in artificial neural networks. Many of them believe that there is some magic secret sauce, that there's some magic learning algorithm that if only they could discover it, suddenly it wouldn't take 1 billion exemplars to train an artificial neural network to do what we do after apparently much less. I think that we have hard-coded essentially hundreds of billions of exemplars over evolution. And so in answer to your question, nothing that we have done so far is helpful. However, one of my main sources of funding right now is IARPA, which is, I don't remember what the other, Intelligence Advanced Research Project Agency. So it's sort of like DARPA, which probably, for all I know, somebody from IARPA is here tonight. But this project that we're working on, Microns, has as its goal to figure out the detailed wiring diagram at the single neuron level of a little chunk of cortex with the goal of abstracting what is essential about that and incorporating it into next generation machine intelligence. So, and we're on a pretty tight timetable. So we have, what is it, October? About 9 more months.
Good luck.
Thank you.
Are you going to do it?
Yep.
Okay.
David Rosen, lifetime member. Question, I was reading that aside from the connectivity, there are certain pathways which are strengthened, the signal strength. Now, isn't that a good part of, that would have to be a good part of what we are outside of even what the connections are. So even if you copy all the connections, would that be a self without the?
Yeah, again, that's a great question. So here's how I think about it. The brain has 100 billion synapses, 100 trillion synapses. We would be a long way toward understanding what's going on if I could tell you, so each individual neuron has about, so let's stick with the mouse. Each mouse cortical neuron selects 1,000 out of 10 million partners to make synaptic connections with, okay? We would be a long way to understanding what's going on if I could tell you which 1,000 of those 10 million possible connections are the ones that individual neurons make. However, there's no question, I mean, I spent my postdoc, so first question was summarizing my graduate work and why I decided that, although that was interesting, that's not the answer. My postdoc was spent studying the processes by which synapses get stronger or weaker. And again, super important, but then if you like, the amount of information encoded or required to specify who's talking to whom is so much greater than the few sort of levels of strength remaining. Well, you needed to describe how strong a connection is, that I think we'd be well on our way to understanding things if we knew that. That said, there's a way to, there's another sort of side project that we're working on that extends this approach to figuring out not only who's talking to whom, but whether that synapse has recently gotten stronger. And I'd be happy to tell you about it, but it's a bunch of molecular tricks. And it's seeming to work, but you know, it doesn't work yet.
Do you no, I guess you have the microphone over here. And then the other guy who stood up, and then if you'll give the microphone to the man with the beard over there.
I think you're on.
But you're next. Yeah, you got it. Is the green light on? He turned it off.
James Griffin member.
There we go.
And Mike, I come from the. Should I press the button?
No, you're fine. Just
That's fine. Hold it to my mouth. Good. It's very close. Okay. And I can blow your ears out. I'm a digital computer kind of guy. My experience has been in physics. And I always thought of these neurons as transmitting electrical signals. So I was astonished tonight to realize there really are chemical tubes carrying molecules of RNA and DNA around. And so, but the question that I would really like to hear from your experience is, how do you compare these brain elements with elements of a digital computing system? And for example, is a neuron like a server, or is it like an electrical junction?
Right. So I didn't really, I should have spent more time talking about how neurons work. So here is one neuron. There's the cell body, and the signals on a fast time scale that neurons use to communicate with one another are largely electrical. All right, so they are indeed, the thing that's making my mouth move as much as electrical impulses coming from somewhere up here and going and controlling my tongue, and that's electrical timescales. There's a chemical step involved that's not really relevant to how synapses work, but it's mostly an electrical signal. That's happening on a timescale of milliseconds. When I was talking about the RNA moving, that's on a much slower timescale of days, and normally neurons don't even drag a lot of RNA. They drag some, but not a lot. We're hijacking the sort of endogenous RNA mechanism to fill the neuron with RNA. So the RNA, the barcodes that were moving around the neuron aren't helping the neuron at all. In fact, they are about to kill the neuron before we remove it. So the way that circuits normally work is they, just as you imagine, they communicate with electrical synapses, with electrical activity. Each neuron receives input from 1,000 other neurons, some of which are excitatory, meaning that they promote activity, others of which are inhibitory, meaning they suppress activity. And if you like, roughly speaking, they like a good model, a good sort of first order model of how neurons work is integrate and fire. So they integrate activity, pluses and minuses, over maybe 5 or 10 milliseconds, and then they either send out a signal or they don't, and then they reset. So I hope that clarified.
So we have 400 billion servers in our brain.
More like each one. I would think each one, I mean, it's closer to a transistor or a logic gate rather than a whole server.
okay, we have a question there.
Hello, my name is Sharif Fahmy, and actually I was going to ask about this slide because you went over it really quickly.
Yeah.
So I was wondering what kind of functional imaging could you use in conjunction with the sort of anatomical stuff?
I put this is a 2-photon image, but I actually stole it from the web because I didn't have an appropriate one for this talk. We do this in my lab, but this happens not to, I don't even remember where I got it from. But they look kind of like this. I apologize for that. But okay, so what can we do? What we can do and what we are doing is we can take a mouse, have it perform a task similar to the one that I showed you at the beginning with a rat. We can image, we do a craniotomy, we put a little piece of glass there. We can image individual neurons, thousands of them at a time, image their activity while the animal is performing the task. And what we do is we find that some neurons become active when the animal hears a particular sound and not another. Other neurons become active when the animal makes the decision to go this way and not that way. Other neurons do more complicated, it's always like that in biology, they always do more complicated things. But our hope is to take those images and now combine those with the in situ sequencing images, these, and say, remember that neuron that we recorded that fired when the neuron was, whatever the neuron heard Beethoven's Fifth? Well, that neuron, interestingly, or this neuron, sends information to the striatum, which is important in decision making. And the reason the mouse appreciates Beethoven's Fifth is because of a circuit that follows from the striatum. That's our dream. That's where we're going with this.
Is the resolution there to be able to identify a specific neuron from both types of imaging?
Yeah, at this point, it's a technical problem. You know, it almost works. There's no fundamental limitation. It's well within the range of things that ought to be doable. Like the stuff that I was telling you about, like, you know, for years I was sort of spending 10 minutes at the end of a talk saying, this is what I'm planning to do. And people sort of shook their head and say, dream on. And so there was absolutely no guarantee that that was going to work. And the fact that it now works is like hugely exciting for me and now for all my detractors. But this, although I said somebody was telling me a story at dinner that they gave a talk and then halfway through the talk they said something so upsettingly controversial that everyone walked out. That is something I strive for. I would love to say something. I haven't achieved that level of success yet. So no, but it's work, but it's not rocket science. It's brain surgery.
So we have a question in the front here because the microphone's there. But the next person is going to be the gentleman with the beard right by the door. If you'll raise your hand, you'll get the mic, and you'll be the next question. Right now, Carl is going to ask a question.
Hi, Carl Merrill. I'm a member of the Society. First, I have to congratulate and thank you. That was a wonderful lecture, and I kept thinking of Boltzmann, who thought, you know, if everything's made of atoms, then atoms can think. And in a way, you know, you've proven that. But one question I have is, Descartes, I guess, who's made the transparent brain by washing out all the fats and everything. And I was thinking you could probably use your tags and then do hybridization with a quantum dot attached to a specific thing and then follow the whole neuron without slicing it into pieces.
Yeah. That's almost the idea that we're pursuing. So Carl Descartes, who's going to win a Nobel Prize soon for a discovery that transformed neuroscience called channel rhodopsin, figured out a method called clarity for clearing out brain tissue and making it transparent. We're actually collaborating with the other person who is going to win a Nobel Prize for channel rhodopsin, named Ed Boyden, who came up with an incredibly clever technique for expanding brain tissue isotropically. He's a fantastic scientist, and he came up with this incredibly clever technique where it turns out that the stuff that they use to make diapers, literally the stuff that they use to make diapers, when you add water, so for those of you who had children who wore modern diapers, they have an incredible capacity to expand. And that same stuff, if you sprinkle it the right way in brain tissue, it pulls molecules apart in a way that makes it possible to use conventional light microscopy to visualize things that previously required super resolution microscopy. And those techniques sort of fit better with this particular technology than Descartes. But yeah, we're right on the same page there.
Yes, now it's your turn. Yeah.
My name is Bruce Muri, and my knees are not cooperating today. I am not a member of the Society, but I do come to a number of these lectures, and I hope to come to many more. My question was this: when you're injecting this label of RNA into the neuron, do you know that it doesn't affect the functioning of the neuron that you're trying to examine? I think that's amazing. But
well, it does.
Yeah.
So
it does.
No, it's a great question, and I skipped over that and many other technical details. So there are a bunch of different viruses that you can use for all sorts of different purposes. This particular virus called Synbis is incredibly toxic. It's an arbovirus, and it kills creatures in no time flat. It expresses at super high levels, but we used it because it's really easy to work with, and it expresses very quickly in 2 days. So in our experiments, in the experiments I showed you, we're only interested in what's connected to what. And we're pretty confident that there's not going to be massive rewiring of the brain on the timescale of 2 days. So for the questions that we're interested in, SynBis is a completely reasonable method for going forward. On the other hand, for other kinds of experiments, where sort of subtle, like this kind of experiment, where a subtle function is important, this kind of function, which is the activity on a timescale of milliseconds, is very sensitive, easily disrupted. And we would be reluctant to do the experiments that we're doing with SynBis and combining them with these experiments. That said, there are other viruses that are available and we and others in neuroscience use. The sort of most commonly used one these days is AAV, adeno-associated virus. It's widely used. We and many others, including Carl Deiseroth, use it. And it turns out to be, there have been many experiments asking whether the delivery of a transgene by AAV disrupts function. And the answer is, in general, no. So, you know, you can't prove a negative. I can't prove that it doesn't change things, but you know, people have been using it in the field for a decade and more, and for at least many things, it's probably fine. And it's also, I think, the leading vector of choice for human gene therapy.
So I think we have a question back here and then a question up here. So, yes. It should be on.
And I am not a member. And I had a question about how do you target the specific barcode that you're looking for in everything else that's going on in the cells? And then could you also use that same technique to target, say, where the cell is exporting serotonin to? So looking at one cell and where it sends the serotonin to in the other areas of the brain.
So do you mean when we target it, you mean how do we sequence just the barcode and not something else?
Yes.
That's a question. We know the sequence. We basically exploit some tricks from molecular biology. We know what the constant regions of the barcode are, and we can put our primers in that target those and not other things.
So you're doing PCR then with
yeah, well, yes, essentially. There's a PCR step that is embedded in there. We also use targeted reverse transcription. But the question of, so I think what you're asking, I glossed over this, is can we combine that with information about what genes a cell expresses? And indeed, that's a fantastic sort of area that we're just starting to move into, because really we would, you know, I think that connectivity is super important, but we also know that there are many different cell types in the brain, some of which express, some of which are, you know, release glutamate, some release GABA, some release serotonin, some have serotonin receptors of one sort, some have serotonin receptors of another sort, and those are all really important to function, and we are working, it's clear how to move forward. We're working on developing those techniques, and I would be working on a grant for one of them right now if I weren't talking to you. It's due in a week. So yeah, again, it's doable, and that's on the table. So we would like what we would call the annotated connectome, where we know not only who's talking to whom, but we know a little bit about each of the players involved.
I think we'll take 2 more questions. We have one right there, yeah. Well, 3. One over here, and then in the back, you'll be the last question.
My name is Jim McCormick, and I'm a member. In fact, as of today, I'm no longer in arrears on my dues.
Yay!
Thank you again for a wonderful, provocative presentation. I have a million questions, but don't worry, I'm going to pick one. And I'd like to go high level and pull the thread on your job security. So I'm very humbled by the scale of what you're trying to do, the years and effort that goes into the very humble steps forward that you're taking. And I guess I'd like to get your opinion on the relationship in your laboratory and the relative value to your ability to move the ball forward between the time you spend doing original research and the time that you spend integrating the research that everybody else in the community is doing. And in particular, I'd like to end up at the point where there's really some value in what you do yourself because you know it better than you could ever get by reading the other guy's paper or even talking to him. Does that make sense?
Is that a yes/no question?
I would ask you to describe the relationship between the unique research you do in your lab and the time you have to spend keeping up with what everybody else is doing so that you can do that unique research.
So what drove me to do this was that, I did not, so there's a great deal of irony in the fact that I am doing this work because I am sort of starting a field of molecular neuroanatomy. And I failed neuroanatomy. I hate neuroanatomy. I literally failed neuroanatomy in medical school. I had to take it over again. And I knew absolutely no molecular biology when I started this project 6 years ago. So literally none. In fact, when I started, I had the idea of doing it, and then it's really, there's a reason I'm advancing here, because it was actually because I used to go running with my former colleague Josh Dubnow, who's a great molecular biologist, and we would go running every day. He'd give me an hour-long tutorial on molecular biology as he sort of helped me formulate and crystallize these ideas. So if the question is, why would I go and try to figure out the connectome when, in fact, a lot of people have figured out a lot of the connections and have published it? The answer is that the way that neuroanatomy is traditionally done, it's sort of it's a labor of love. I showed you those 25 neurons from 28 rats, and there's no database where you can go and figure out what those 25 neurons from the 28 rats do. You have to go and look at the pictures and read their loving description. And if you hear neuroanatomists talk about their neurons, I mean, they're like tactile experiences. There are some really beautiful and it's wonderful that they feel that way, but I just want to know who's talking to whom, and that information is not available. So I'm from my point of view, I just want I want to be able to sit down at a computer and go, oh no, that idea doesn't pan out. And so I spend a lot of time. So I don't spend enough time reading the neuroanatomical literature because I can't. I try to get other people to read it for me. I don't have that love of the curves of each particular axon as it courses through the striatum. But what I do read all the time are the newest, latest advances in molecular biology, because that is a field that is, somebody pointed out at a meeting that in neuroscience we still show pictures from Ramon y Cajal, who is the granddaddy of neuroscience, but we don't show them for historical reasons. We show them because he got the answer right, and we haven't moved beyond that. Like no one's done better, right? And so, I didn't want to be in a field, where things haven't moved forward in 100 years. Molecular biology, if you haven't read the papers in the last 2 years, you're out of the game. So I don't know if that answered your question, but
yeah.
Hi, my name's James Fraser. I'm sorry, I'm not a member. To follow
Can you
up on
hand off the microphone to the next person?
It's easy to view.
Sorry.
No, I don't know.
Wow, okay, life memory
I'll join. But, to follow up on the question from the gentleman over here, it seems as if you are mapping the movement of DNA or RNA throughout the brain, as the neurons are moving, connected to others.
I'm tricking the neurons into moving RNA so that I can map it. They don't normally do that.
Okay. And but that is different somehow than the electrical signals that when I'm talking or I'm thinking about the election or something like that, that's electrical signals moving around.
Yep.
How do we know or do we know that those two are the same map, or are these different levels of functioning? There's a rational functioning and you're doing a map of our emotional functioning?
No,
or is that not the way to think about it?
The way I think about it is that the neurons, the pathways, are like roads, and if you like, the electrical signals are like individual cars that travel down those roads. All right, so unless you have an off-road vehicle, your car has to follow the known roads, and so I'm mapping out the possible routes by which a car, i.e. a neural signal, can get from one place to another. And the way I'm doing it, if you like, is I'm painting each road with a unique identifier. So, we have those in real roads, right? Like if you're driving along, at each point it says you're still on I-95, still on I-95, still on I-95. In the brain, we don't have those I-95 markers. And so, I get lost even with them on roads. But without them, I would be totally lost. And so that's sort of, I would say that's the analogy, that the signals are, happening on the time scale of milliseconds. Those are cars moving really quickly along those roads. And the road, we are basically, if you like, forcing the roads to become unique colors, unique sequences. I hope that helped. I mean, the analogies sort of break down at some point, but there are two completely different timescales.
In the back, stand up.
My name is Terry Layton. I'm a member. I want to go back to that slide you showed of the brain bathed in drugs.
Uh-huh.
For the work you've done so far, is it at the point where anyone has tried to apply it? Or how far in the future is the application to applying it to targeted drug delivery or deep brain stimulation to treat serious mental illness?
Well, so I should say that the data that I showed you here with the rotating brain, we collected in August. So far, that has not resulted in any application. However, work from other people's labs, including Fritz Henn, who's sitting here, on neural circuits underlying disorders like depression, I think have had implications and are having implications for delivery of drugs and for stimulation of specific circuits. So I would say that is the where people are going. I don't know, Fritz, do you want to comment on whether there are specific treatments that have arisen from, for example, I think yours is one of the best examples of. So, yeah, I mean, it's incredibly frustrating how long things take to come to the clinic, but yeah.
So you've got an anatomy which you think may hit the pathology, but there's another level beyond what Tony's actually shown us, and that may play a real role, and that's that synapses come and go, and we have to catch them, whether they're overexpressed or underexpressed. And I don't know whether you can do that, Tony. I've been wondering about
Let's talk about that.
Yeah, that's a real problem. Yeah.
Sure. So I think the answer is that we're getting close to being able to use some of the understanding that we've gotten from circuitry and using that in the clinic. I think some of the more immediate applications will actually come from, have come and are coming from deep brain stimulation. But in the long run, that's pretty invasive. And so, for intractable depression or other disorders, the payoff is worth it. But ideally, we would have a less invasive way of targeting specific circuits. So I think that's where the future is.
Well, thank you. So before you go, I'd like to present you with this small token of appreciation. It's a framed copy of the announcement of your lecture, signed by the members of the General Committee on behalf of all of the members of the Society and the guests here tonight.
Well, thank you.
Thank you. So I have a few closing announcements to make, and before I make them, I have to switch over to the computer and hope that this works. So I have a question, which you can speculate on. So you showed a slide with a few signals, and I think you said that every neuron you looked at had a somewhat different, not signals, but a somewhat different set of connections. So when you talk to your signal processing friends in the sort of distributions, did they see anything that looked like maybe there was some corollary between the connections and signal processing schemes that they're familiar with?
So we're starting to do that a little bit more rigorously from the, I think that the limitation with the data set that we have is that 1,000 neurons just isn't very much. Turned out not to be enough. There just aren't that many neurons in that locus in our spatial resolution of the retina. So we're doubling down now on the cortical link data, and we're finding all sorts of interesting structure. But 6 months, I hopefully will have.
Too early to tell.
What's that?
Too early to tell.
Too early to tell. Okay. There's a huge amount of structure there, but I can't tell you what it is.
Before we adjourn to the social hour, a few closing items. PSW depends on the membership, and without these, no PSW, and especially no free lectures and no free refreshments. So please join if you're not a member. Oh, we're there. Thank goodness. Sometimes my computer decides to wait. It's easy to become a member. This is the home page. You'll notice that there's a membership button, and all you have to do is press on the membership button. And when you do, you will pull up the membership application form, believe it or not. And the membership application form will ask you for very detailed, complicated information, like your name and where you live, and maybe a little bit about your educational background and a few other things, but nothing complicated. There's no IQ tests. There's no hurdles to surmount, no obstacles to overcome. Membership is open to all people with a keen interest in science and in PSW activities. At the bottom of the page, which you can't see here, but you can see here, you'll see there's a little button where you pay your dues. Okay, very important, pay your dues. And I do want to mention, don't be confused. You'll see something about PayPal. It does not mean that you have to have a PayPal account. Every merchant, and in this case we're acting as a merchant, has to have a merchant banking account in order for payments by credit card or debit card to be processed. PayPal offers a very, very low rate to nonprofits. That's why we use PayPal. But you don't have to have a PayPal account. You can use any debit or credit card you want. And so just go to the debit or credit card tab. Don't be confused by the PayPal logo, and pay your dues, and we will then receive your application. We will review it, and we will shortly send you a notice that you are now a member. Except in the 1 in 100,000 applications where we decided maybe you are really just interested in philosophy and not science, and perhaps you should join the Philosophical Society of Philadelphia. If you're a meetup member, please keep in mind being a meetup member is the same as being a PSW member, and please join PSW. Please see our membership chair, James Heelan. I don't know if he's there, he is. Or corresponding secretary, Robin Taylor. If you have any questions about membership, giving us contributions, or getting involved in the Society's activities, remember PSW is a nonprofit educational organization, tax exempt under Section 501c3 of the Internal Revenue Code. It's going to be increasingly important to you probably, and contributions are tax deductible. Our next lecture, no, I'm not, okay. There we go. Our next lecture will be the 2,368th lecture. It will be on November 4th, 2016, right here in the Powell Auditorium. And the speaker will be Tom Cronin, Professor of Biological Sciences at the University of Maryland, Baltimore County. He will be speaking on seeing the light, the wonderful diversity of light perception in living organisms. We hope to see you then for what promises to be a very informative and entertaining lecture. The fall schedule has been posted to the website, www.pswscience.org. Please check there at regular intervals for updates to the schedule. We will be posting the spring schedule in the future, not too distant future. Finally, the social hour ends at 10:30, after which PSW members and their guests meet across the street at the Fairfax Hotel Lounge. If you want to join us, please speak with corresponding secretary Robin Taylor, Vice President Lloyd Bitchell, or me, sometime during the social hour. Please use the side entrances to exit the building, And I will now accept a motion for adjournment of the 2367th meeting of the Society to the social hour. Okay, we have raised hands. Those must be motions. Do we have a second? We have a second. All in favor? All opposed? And the meeting is adjourned to the social hour.